Look at the last medical form you ever filled out. Somewhere on it, there was a small box, and inside that box, you circled a single letter for your blood type. A, B, AB, or O. Most people glance at that letter for half a second and never think about it again. But if the letter you circled was O, you are carrying something inside your own cells right now that reaches back further than your family tree, further than your language, and quite possibly further than our species itself. For most of the last 100 years, science told the story of that letter almost completely backwards. It took over a century, several accidental discoveries, and one very strange piece of ancient DNA to finally get it right. The story does not start with ancestry at all. It starts with a young doctor in Vienna named Karl Landsteiner, working alone in a small laboratory in the year 1901. Landsteiner was not hunting for deep human history. He was trying to solve a much more immediate problem. Doctors at the time knew that blood transfusions sometimes saved a patient's life, and sometimes killed them within minutes. And nobody understood why. It looked less like medicine and more like a coin flip. Landsteiner began mixing blood samples from different people on glass slides just to see what happened. Some samples blended together smoothly. Others clumped into visible little clusters almost immediately, a reaction that meant the two bloods were fighting each other. That single, simple observation cracked open an entire hidden system inside human biology. Landsteiner sorted the samples into groups based on how they reacted, and those groups became the 4 blood types doctors still use today: A, B, AB, and O. The discovery won him a Nobel Prize decades later, and it turned blood transfusion from a dangerous gamble into one of the safest, most routine procedures in modern medicine. Surgeons who once lost patients to reactions they could not explain suddenly had a simple test that could predict, with near certainty, which blood would work and which would kill. Before Landsteiner's discovery, doctors attempting transfusions were essentially guessing with human lives, sometimes mixing blood from an animal or from a stranger with no way of knowing whether the two would fight each other inside a patient's veins. Some patients recovered, others died within minutes, and for a long time, nobody could explain the difference. Landsteiner's simple mixing test changed that almost overnight, and by the middle of the 20th century, blood typing had become a standard part of medicine practiced in hospitals everywhere. For most of the century after that discovery, a blood type was treated as nothing more than a practical label, the kind of thing written on a hospital wristband or a donor card. It told doctors whether a transfusion was safe. It did not, as far as anyone assumed, tell you anything about where you came from, how old your ancestors were, or what they had to survive. That assumption sat mostly unquestioned for decades, right through most of the 20th century, while blood typing quietly became one of the most useful tools in the history of medicine. Nobody was combing through a person's blood type looking for a fossil record. The idea that a single letter on a lab form might function as a kind of biological time capsule, holding information about events that happened hundreds of thousands of years ago, simply was not part of the conversation yet. That gap, the space between what blood typing was assumed to mean and what it actually turned out to hold, is where this story gets interesting. You have probably come across a popular claim online at some point, maybe without even realizing how popular it actually is. The claim goes something like this: Type O is the original human blood type, the ancestral version that our earliest hunter-gatherer ancestors all carried before anything else evolved. It shows up in casual conversation, in old wives' tales passed between relatives, and it has even been used as the foundation for entire diet and lifestyle plans built around the idea that people with type O blood should eat the way ancient humans supposedly ate. It sounds intuitive. It sounds like the kind of thing that should be true. And when geneticists actually went and tested it directly against the DNA, it turned out to be backwards. Here is what researchers actually found. The version of the gene responsible for type A blood is the one shared most broadly across primates, including our closest living relatives like chimpanzees. That makes the A version the older baseline, genetically speaking, not type O. Type O exists because at some point in our evolutionary past, a mutation broke that older gene. In a narrow, technical sense, type O is actually the newer version of the two, not the ancestral one. That single correction does not make the story less interesting. It makes it stranger, because being a newer mutation does not mean being recent, and the gap between those two ideas is exactly where the real history is hiding. To understand why a broken gene can still be almost impossibly old, it helps to know exactly what is physically happening inside your own cells right now. Somewhere on chromosome 9, one specific gene has a single job. It builds a small enzyme, and that enzyme's only task is to attach a particular sugar molecule onto the surface of your red blood cells. Attach one version of that sugar, and the surface of the cell carries the marker for type A. Attach a slightly different sugar, and you get the marker for type B. Carry the instructions to attach both, and you get type AB. Think of that gene like a recipe card handed down to a factory worker inside your body, and the enzyme it produces, like the worker following that recipe exactly, step by step, sugar molecule by sugar molecule. In the most common version of the gene that produces type O blood, a single letter is missing from that recipe card. It sounds like an almost trivial change, one letter out of thousands, but delete a single letter from a written sentence, and everything that comes after it can turn into nonsense because every word downstream shifts out of place. The exact same thing happens inside this gene. The factory worker still shows up for the job. The recipe still gets read. But because one letter is missing, the instructions no longer make biological sense past that point, and the enzyme that gets built comes out broken. No sugar ever gets attached to the surface of the cell at all. A type O red blood cell is essentially a blank surface carrying no flag, no marker. Nothing for an immune system or a pathogen to grab onto. And this is also, in plain terms, why type O blood is often called the universal donor type in emergency medicine. Without those extra sugar markers on the surface, a type O red blood cell has far less chance of triggering an immune reaction when it's given to someone with a different blood type, which is exactly why hospitals keep it in reserve for emergencies when there isn't time to run a full match. So if type O comes from a mutation, and that mutation is technically newer than the gene it broke, why does this story reach back before our species even existed? This is exactly the part where most casual retellings get tangled up, so it is worth slowing down here. Scientists cannot carbon date a single missing letter directly the way they can date a fossil or a piece of charcoal. What they can do instead is study the DNA sitting immediately around that mutation. Every single time a piece of DNA gets copied down through a new generation, tiny additional changes accumulate nearby, almost like growth. Rings building up year after year inside a tree trunk. If you collect DNA samples from people carrying type O blood on every continent on Earth, and count how many of those small extra changes have built up around the same mutation, you can estimate, with real statistical confidence, roughly how long ago that original mutation first appeared. This method has been tested and refined for decades, and it consistently produces the same kind of answer, no matter which population researchers happen to sample. If you have followed the story this far, you already understand more about a single letter on a hospital form than the vast majority of people who have ever circled it without a second thought. And there is a genuinely strange exception coming up later in this video, a case rare enough that it makes even experienced geneticists pause, so stay with this. When researchers actually ran that dating analysis on the mutation behind type O blood, the number that came back was almost too old to take seriously at first. It did not simply predate the migration of modern humans out of Africa, an event that most genetic evidence places somewhere between 50,000 and 70,000 years ago. It went back dramatically further than that. When scientists sequenced ancient DNA extracted from Neanderthal skeletons recovered at cave sites in Spain, they specifically checked whether Neanderthals carried a related version of the same broken gene. They did. A separate analysis of Denisovan remains recovered from a cave in Siberia turned up related variants as well, sitting inside DNA from an entirely different archaic human population on a different continent. Think carefully about what that actually means. This mutation was not something that appeared sometime during modern human history, after our species had already taken shape. It existed inside an ancestral population before modern humans and Neanderthals had even split apart from each other as separate lineages, which pushes the minimum possible age of this mutation into the hundreds of thousands of years, long before anyone who looked or thought the way we do today ever existed to carry it forward. Zoom out even further, past that split between humans and Neanderthals, and the broader family of genes responsible for blood type in general. Traces back tens of millions of years into ancestry we share with primates far more broadly, long before anything resembling a human walked anywhere on Earth. The specific single-letter deletion that causes modern type O blood is younger than that much deeper split, but it is still, by every measure researchers have tested, almost certainly older than Homo sapiens as a species. That is not a small or casual claim. It means the exact letter you might have circled on a medical form last year is a living fragment of a mutation first carried by ancient hominins, creatures who had no concept of medicine, no written language, no idea that anything they carried inside their blood would ever matter to anyone. And somehow, without a single gap, that fragment has been copied forward, generation after generation, in an unbroken chain that reaches all the way to you. A claim this large should not rest on just one gene, and thankfully it does not. If type O blood were the only piece of evidence pointing toward deep ancestry, a reasonable person could call it a fluke, one unusual result from one small data set. What makes the story solid is that the exact same pattern shows up again and again, independently, across completely different, unrelated parts of the human genome. Researchers studying the Y chromosome, the piece of DNA passed almost unchanged from father to son, have found ancient branch points reaching back hundreds of thousands of years. Researchers studying mitochondrial DNA, passed almost unchanged from mother to child, have found the same kind of pattern on a completely separate part of the genome. None of these systems are structurally connected to the gene behind blood type. They were studied by different research teams, working decades apart, using different tools, and they all keep pointing back toward the same general place and the same general timescale. That kind of independent agreement between data sets that never spoke to one another is exactly the kind of evidence that convinces scientists a pattern is real instead of a coincidence. Some of the clearest evidence for exactly how old this all really is comes from tracing two very specific pieces of DNA back through time. Every man alive today carries a Y chromosome, passed down almost unchanged from father to son across an unbroken chain of generations, which makes it a remarkably clean tool for tracing paternal ancestry. Compare Y chromosomes from men sampled all over the world, and researchers can trace every single one of them back to a shared convergence point, sometimes given the nickname Y chromosomal atom. That name is a little misleading because it does not refer to one literal first man. It simply marks the point where every living man's paternal line eventually meets, statistically speaking. There is a mirror version of this story on the maternal side. Mitochondrial DNA passes almost unchanged from mother to child, making it the maternal counterpart to the Y chromosome and a completely independent way to trace the deepest roots of the human family tree. When researchers build that maternal tree out, the very oldest branch consistently traces back to populations in southern Africa. Every mitochondrial lineage found anywhere else on Earth, in Europe, in Asia, across the Americas, or throughout the Pacific, nests inside that African branch as a smaller, later offshoot. Both of these separate systems, one following fathers, one following mothers, keep landing on the same general conclusion using completely different genetic material, which is exactly the kind of independent confirmation that makes geneticists confident this is not a coincidence. There's a second, completely separate line of evidence that lines up with all of this, and it comes from population genetics rather than any single gene. Every population living outside Africa today, every European, every East Asian, every indigenous American, every Pacific Islander descends from a relatively small group of people who left the African continent sometime in roughly the last 50 to 70,000 years. When a small group splits away from a much larger population, they only carry forward a fraction of that original population's full genetic diversity. Geneticists call this a founder effect, and it compounds every single time it happens again, through every later migration into new territory. The practical result is one of the most consistently repeated findings in the entire field of human genetics. The farther back you trace a population's ancestors from East Africa, the less overall genetic diversity the population tends to carry today. The number this produces is honestly hard to believe the first time you hear it. Two people sampled from different populations within Africa can differ from each other genetically more than someone from Norway differs from someone in Japan. That is not a rounding error or a strange coincidence. It is the fingerprint left behind by the simple fact that modern humans have lived in Africa far longer than anywhere else on Earth, long enough for far more genetic diversity to build up there before any of it ever traveled elsewhere. The fossil record backs this up in its own way. For most of the 20th century, the oldest confirmed Homo sapiens fossils were dated to roughly 195,000 years old, discovered in Ethiopia. Then, in 2017, a research team returned to a set of bones originally pulled from the ground in Morocco back in 1961, bones that had been filed away for decades under the assumption that they belonged to a fairly recent Neanderthal. Using modern dating techniques, the team found those bones were actually around 300,000 years old, with a face that looked unmistakably modern, even though parts of the skull behind it were more archaic in shape. It was not a strange, isolated fluke. It was evidence that our species did not appear suddenly in one place, in one moment, like flipping a switch. We took shape gradually, across multiple regions of Africa, over a much longer stretch of time than most people ever learned in school. Put the fossil evidence next to the genetic evidence, and the two tell the same basic story from two completely different directions. Something like 80 to 85% of our species' entire existence happened inside Africa, long before a single group ever crossed into Asia or Europe. Everything that happened after that migration, every civilization most history classes spend the most time on, is not really the main chapter of the story. It is closer to the epilogue. This is where the story stops being only about one gene and becomes something with real, measurable stakes. Because your blood does not just carry old history. It carries a record of what your ancestors had to survive in order for you to exist at all. Map blood type frequencies across the modern world today, and clear geographic patterns jump out almost immediately. Type A runs unusually high through parts of Western Europe. Type B is especially common across northern India and Central Asia. Type O dominates large stretches of West Africa. And it is also extremely common among indigenous peoples of the Americas, for reasons tied to a separate, much more recent wave of migration. Some of that pattern connects directly to one of the deadliest threats human beings have ever faced across our entire history as a species: malaria. Several independent studies have found that people with type O blood tend to develop noticeably less severe cases of the deadliest strain of malaria compared to people carrying types A, B, or AB. The leading explanation is that infected red blood cells clump together less easily when they are missing those extra surface markers, which appears to slow the disease down inside the body. It is worth being completely honest here, because an honest telling of this story matters more than a dramatic one. This particular link between type O blood and reduced malaria severity is still an active, genuinely debated area of research. The size of the protective effect shifts from study to study and from population to population. It is not yet a fully settled fact, the way some of the other evidence in this video is. A much more clear-cut example sits right next to it, involving a different, less familiar blood marker called the Duffy antigen. Most people of European and Asian descent carry a specific molecule on the surface of their red blood cells, one that has nothing structurally to do with the A, B, or O system. But among people of West and Central African descent, something close to 90 to 95% carry no version of that molecule at all. It is one of the sharpest, most clearly defined genetic dividing lines found anywhere on the entire planet, nearly universal in African and African-descended populations, and rare almost everywhere else. That difference is not a coincidence, and it is not random. A specific strain of malaria parasite needs to grab onto that exact molecule in order to invade a red blood cell successfully, the same way a specific key needs a matching lock to open a door. Remove the lock entirely, and the parasite has nowhere left to attach itself. Across huge regions of West and Central Africa, this particular strain of malaria has nearly disappeared over time, not through medicine or chance, but because the population living there evolved over an enormous span of generations to remove the exact biological door that the parasite depended on to survive. If you laid a map of this trait over a historical map of that specific strain of malaria, the two maps would practically trace each other, dense in the exact regions where the disease was most severe and thinning out rapidly the further you move away from that zone. Duffy and type O are not even the entire toolkit your ancestors' bodies built up against malaria over time. There is the sickle cell trait, which subtly reshapes red blood cells in a way that makes it much harder for the malaria parasite to survive and multiply inside them. There is a variant related to a condition called G6PD deficiency with a broadly similar protective effect. Each of these traits comes bundled with a real trade-off, which is part of what makes them so scientifically interesting. Carry a single copy of the sickle cell variant, for example, and you generally receive strong protection against the most severe forms of malaria. Carry 2 copies instead of 1, and you develop sickle cell disease, a serious condition in its own right. That built-in trade-off is exactly what keeps these genes circulating steadily inside a population across thousands of years, instead of either disappearing completely or spreading to take over the entire gene pool. Stack all of these separate mechanisms together, the broken sugar molecule behind type O, the missing lock behind Duffy, the reshaped cells behind sickle cell, and you are looking at an entire evolutionary arsenal built up gradually over an enormous stretch of time against one of the single deadliest diseases our species has ever faced. Your blood does not just remember roughly where your ancestors lived on a map. It remembers, in very concrete biological terms, what they had to survive to get you here today. This is exactly why genetic ancestry testing has become so emotionally significant for so many families, especially for descendants of people who were forcibly taken from Africa during the transatlantic slave trade. Historians estimate that more than 12 million Africans were forcibly removed from their homes during that period, with roughly 10.5 million surviving the crossing itself. The journey, often called the Middle Passage, could take 1 to 3 months, under conditions deliberately designed to strip away every trace of a person's individual identity before a ship ever reached land. Names were erased, languages were stripped away, families and communities were split apart, often on purpose as a method of control. The paper trail that should have preserved generations of family history was destroyed, falsified, or simply never written down in the first place. For countless descendants today, there is no surviving document that leads back to a specific place, a specific people, or a specific person. For a long time, that felt like a door closed permanently. It is exactly why it means something real, when a modern DNA test can compare someone's genetic markers against reference populations, collected from around the world, and say, with genuine statistical confidence, that a lineage traces most closely to a particular region or community in West or Central Africa. That is not a small thing to hand someone whose family history was deliberately erased 2, 3, or 400 years ago. It is a name appearing where there used to be only blank space on a form. Companies offering consumer DNA testing today are built on the same underlying science described throughout this video, reading small genetic markers, sometimes including blood type variants, against enormous reference databases collected from populations on every continent. Most results will not turn up something as dramatic as a mutation tracing back before our species existed. But the underlying method is exactly the same careful counting of small genetic changes, applied quietly to ordinary people every single time someone spits into a tube and mails it away, hoping for a clearer picture of where they actually come from. If a story this old and this personal has grabbed your attention so far, it is worth sticking around, because there is one more twist coming that manages to complicate almost everything you have just heard in the best possible way. Every so often, a field of science that feels mostly settled turns up an exception strange enough to make researchers stop and reconsider what they thought they understood. Blood type has one of the strangest exceptions on record, and it is called the Bombay phenotype. In extraordinarily rare cases, a person can inherit 2 copies of a completely different, unrelated broken gene, one that sits earlier in the same biological pathway than the gene responsible for types A, B, and O. This particular mutation prevents any surface marker from ever forming. On the red blood cell at all, not the marker for type A, not the marker for type B, and not even the blank, flag-free surface of standard type O blood. People with this condition are sometimes described as having blood so rare that it does not fit cleanly into the ordinary 4-type system most of us learned about in school. It was first identified in the city of Bombay, now known as Mumbai, which is where the name comes from. Fewer than a few hundred confirmed cases exist anywhere in the world, spread thinly across different populations, and people who carry this trait can typically only safely receive blood donations from someone else who shares that exact same extremely rare condition. In a genuine medical emergency, finding a compatible donor can become extraordinarily difficult, sometimes requiring donor networks that span entire countries, and in some documented cases, patients have had blood flown in from donor registries on other continents just to find a safe match. Geneticists studying this condition point out that it does not fit neatly into the same evolutionary story as ordinary type O blood, since it arises from an entirely different broken gene earlier in the same biological pathway. It exists as its own separate reminder that biology rarely stays as simple as a 4-letter chart taped to a hospital wall. It is a small, quiet reminder that even as system scientists have studied carefully for well over a century can still hold real surprises, hiding in plain sight inside a small number of people alive today. So here, after all of that, is what the evidence actually supports, laid out plainly alongside what it honestly does not. Type O blood comes from a mutation that broke an older, more ancient gene. A mutation old enough to predate our own species entirely, and old enough to show up clearly in the DNA of Neanderthals recovered from ancient cave sites on a different continent. Its modern global patterns connect, at least in meaningful part, to a long evolutionary battle against malaria, working alongside other genetic defenses like the Duffy antigen and the sickle cell trait. What all of this evidence does not do, and this matters, is predict your personality, your intelligence, your specific ancestry on its own without other supporting evidence, or anything at all about your destiny as a person. Any account of blood type science that claims otherwise, no matter how confidently it is presented, has wandered well past what the actual research can honestly support, and it deserves to be treated with real skepticism. What all of this really adds up to is not a single dramatic fact, but a layered, carefully checked picture built from a broken gene, ancient bones, and generations of careful counting. What is left, once you strip all of the exaggeration and the myths away, is something quieter than a dramatic headline, and arguably far more remarkable. It is a single letter on a form connecting you right now, in this exact moment, to a mutation that existed before modern humans did, carried forward without interruption by an unbroken chain of ancestors who had no possible way of knowing what they were passing down. So the next time you glance at that letter on a medical chart, or think seriously about where your own biology actually comes from, remember what is genuinely sitting behind it. A fragment of deep evolutionary history, quietly being carried forward, one person at a time, through you. If a single letter on a form can hold a story reaching back this far, it might be worth wondering what else your own DNA has been quietly holding on to this entire time, waiting for someone to finally ask the right question. If this kind of hidden history inside your own biology caught your attention even for a moment, there's a lot more of it waiting to be uncovered. So stick around and subscribe for the next one.